Dear friends, welcome back. Let us listen to the viral insecticides, which is one of the biological pesticide. Microbial insecticides are a new form of a pesticide that work by infecting the selected insect populations with the bacteria, viruses, or fungi. These are comprised of microscopic living organisms, that is the bacteria, fungi, protozoan, or nematodes, or the toxins produced by these organisms. They are formulated to be applied as conventional insecticidal sprays, dust, liquid drenches, liquid concentrates, wettable powders, or granules. Each product each product's specific properties determine the ways in which it can be used most effectively. Coming to the viral insecticides, viruses are potentially valuable tools for managing pest insect population. Their host specificity makes them compatible with other natural enemies and are safe to use in environmentally sensitive areas. Viruses that are pathogenic for insects are found in the families Bacloviridae, Poxviridae, Rheoviridae, and some others. The most frequently studied of these viruses are the nuclear polyhedrosis virus, granulosis viruses. The family Bacloviridae includes the nuclear polyhedrosis viruses and granulosis viruses. Nuclear polyhedrosis viruses in which
Announcement for the CMA test.
the most frequently studied of these viruses are the nuclear polyhedrosis viruses and the granulosis viruses. The family Bacloviridae includes the nuclear polyhedrosis viruses, that is NPV, and granulosis viruses, GB. Nuclear polyhedrosis viruses, in which numerous viruses particles are packaged together in a crystalline envelope within insect cell nuclei, whereas granulosis viruses, which are one or two virus particles, are surrounded by a granular or capsule-like protein, which is crystal found in the host cell nuclei. Coming to the nuclear polyhedrosis viruses, the example is baculovirus. Baculovirus are pathogens that attack insects and other arthropods. They are composed of double stranded DNA that codes for the genes needed for viruses establishment and replication. Because this genetic material is easily destroyed by exposure to sunlight or by conditions in the host gut, an effective baculovirus particle, that is a virion, is protected by protein coat called as polyhedron. The new NPV from Autographica, California, is one of the most intensively studied species. The infectious virus particles or virions of NPVs can be enveloped singly or multiply, that is SNPV type or MNPV type, and are occurred in protein bodies called polyhedra. NPV polyhedra may contain few or many virions. After ingestion by the host, that is insect, and reproduction in the midgut of the cells of the insects, other tissues and organs in the insect become infected, primarily the fat body, epidermis, and blood cells. This picture shows the electron microscopic structure of baculovirus, that is polyhedrosis virus. The polyhedron virus enters the cell and where it forms the nucleocapsid and a virion particle inside the cells, epithelial cells of the insects. Coming to the mode of action, the baculovirus infection involves two distinct forms of viruses. One is occlusion derived viruses is present in the protein matrix, that is polyhedrin or granulin, and is responsible for the primary infection of the host. The other one is budded virus is released from the infected host cells later during the secondary infection. Here you can able to see the budded virus, occult virus and occlusion bodies of the baculovirus. The initial infection occurs when a susceptible host insect feeds on the plants that are contaminated with the occulted form of the virus. Next step is once the ocult bodies reaches the midgut of the insect, the alkaline pH causes the dissolution of the occlusion body, releasing the virions, that is occlusion uh, dissolution virion. The released virions then pass through the peritrophic membrane of the midgut of the insect. Once the virions have entered the midgut of the epithelia of the insect, the nucleocapsids migrate into the nucleus of the cells. The next step is this migration may be in association with the cellular actin. Once the nucleocapsids reach the nucleus, the DNA is uncoated into the nucleus. Next one is the virus transcription and replication occurs in the cell nucleus and new BV proteins, BV particles are budded out from the basolateral side to spread the infection systematically. During budding, the baculovirus acquires loosely fitting host cell membrane with expressed and displayed viral glycoproteins. Here is a picture shows how the viral particles enters into the epithelial membrane of the gut of the insects, then it reaches the cell and then it multiplies rapidly and makes the insect die. NPV infection of an insect host. Okay, you're able to see the injection of the polyhedrosis virus by the mouth part of the insects. Then it reaches the foregut, and then in the alkaline pH, the occlusion bodies are dissolved, and in the midgut, the DNA are released and it is multiplied in large, and at the peritrophic membrane it multiplies and more and makes the insect paralysis. In the phase 1, as soon as the entry of the insect, expression of viral transregulators and the genes which do not require transregulators for efficient transcription. Many of the genes expressed in this phase are involved in establishing the infection. In the delayed early stage, the expression of genes involved in the replication of the viruses and the manipulation of the host. The delayed early genes often require the presence of viral transregulators, that is IE0, IE1, P38 for efficient transcription. Coming to the late phase, during the late phase, transmission from early to late is characterized by shutdown of the host cell, DNA replication, and protein synthesis. Nucleocapsids are produced, budded viruses is produced and disseminates the virus throughout the host. I used to say to my students that the hijacking activity of the activity of the viruses over the host cell. The viral DNA hijacks the host cell, so host cell will not produce its own DNA. Instead, it replicates and multiplies the viral DNA and viral protein. And coming to the very late period of infection, it is advanced stage of viruses infection. Virions become occluded on the protein polyhedron, and viral proteases liquefy the host and liquefy the host and degrade the chitinous exoskeleton. Occult progeny virus is disseminated onto surrounding material for horizontal spread. Infection with the baculovirus is historically called wilting disease because the tissues of the host liquefy and infection of the epidermis causes the host to appear to melt, releasing virus particles into the environment. Production of nuclear polyhedrosis viruses by using field-collected larvae of Helicoverpa. As you all aware, the viruses are obligate intracellular parasite. It cannot be cultured under laboratory. Hence, the insects are used for the multiplication of the viruses. Helicoverpa armigera is the most pest of the different crops. The pest is active throughout the year. The nuclear polyhedrosis viruses has got occult, that is a rod shaped singly or in groups in polyhedral occultation bodies. Site of multiplication is cell nucleus of epidermis, fat body, blood cells and trachea. Collection of insects. Healthy larvae should be collected in cell walls from any infested fields. For more HNPV yield, larvae selected for production should be fourth instar. Insect rearing can be done in optimum rearing conditions such as 24 to 36 degrees Celsius of temperature are required for HNPV production. Food should be available at all the times. Cleaning the trays and equipments before rearing is necessary to avoid the buildup of contamination by protozoans and also bacteria. To prevent oviposition by flies on dead larvae. Inoculation of viruses. The larvae are supplied with well-soaked 2 to 3 virus inoculated chickpea seeds. Care should be taken to thoroughly mix the inoculum with the chickpea seeds. Next one is the virus harvesting. The infected larvae to die naturally in 4 to 6 days. The virus completes its life cycle to achieve maximum virus production. Larvae with clear syndromes of NPV should be collected at soon after death, leaving those infected with other pathogens. Larvae become pinkish white, especially in the ventral side, due to accumulation of polyhedra. In advanced stage, the larvae become flaccid, the skin becomes very fragile and eventually rupture. Processing of the viruses. Blend the dead larvae in the suitable blender to crush the insect tissue and release the NPV. Collect the material, washing with the distilled water and filter through muslin cloth to remove the remaining parts of the larvae body. The solution is centrifuged at 2500 rpm for 15 minutes to separate the virus from homogeneous liquid. After centrifugation, the liquid, which is turbid and loose on the top of the tube, is poured off, and the paste-like substances at the bottom, which full of NPV, is saved. Measuring the quality of viruses. Measure the quality of viruses by hemocytometer. Either dark field or phase contrast microscope is needed to count the polyhedral inclusion bodies using a hemocytometer. Applications. Spraying the NPV late in the day after peak sunshine to improve the effectiveness of the NPV. Relative effectiveness. It is widely acknowledged that baculovirus can be as effective as pesticides in controlling specific insect pests. However, the expense of treating a hectare of land with a baculovirus product invariably costs more than an equally efficacious chemical treatment. This difference in price is due primarily to the labor-intensive nature of baculovirus production. Some viruses can be produced in vitro within cell cultures. The laboratory is not requiring whole or living insects. These are less expensive than those that can only be produced in vitro, that is, inside the living insects. Pest attacked by NPV. Nuclear polyhedrosis viruses affects various insects like alfalfa looper, corn earworm, imported cabbage worm, cabbage looper, cotton bollworm, cotton leafworm, tobacco budworm, army worms. European cork borer, almond moth, spruce budworm, Douglas fir, tussock moth, pine sawfly, and a gypsy moth. Coming to the last part is preparation of the granulosis viruses have been isolated from several caterpillar species, including imported cabbage worm, cabbage looper, army worm, um, fall webworm, and mosquitoes, among many others. Here you can able to see the picture of cabbage looper larvae infected and killed by nuclear polyhedrosis viruses. Okay, you can able to see the live worm. You can able to see the infected nuclear polyhedrosis viruses that kill the worm. Appearance of the nuclear polyhedrosis viruses. Insects killed by baculoviruses have a characteristic shiny, oily appearance. They are extremely fragile to the touch, rupturing to release fluid filled with infective viruses particles. Coming to the habitat, baculovirus can be found wherever insect exists because rain and wind readily carry baculoviruses from place to place. It is likely that every piece of land and a body of water contains some virus particles. Advantages of using virus biopesticide. Baculovirus have a number of unique features that have generated interest in their use as a biological insecticide. They include high pathogenicity and virulence, limited host range, that means very low risk to non-target insects. Absence of toxic residues on fruits and vegetables. Easily applied using conventional spray equipments. Long shelf life, that means it can be alive, activate for several long years. Granulosis viruses. Granulosis viruses are closely related to the nuclear polyhedrosis viruses and are similar in structure and pathogenesis. The major differences between these two groups is that the virions are singly occuled into small occultation bodies called granules. Like the NPVs, the reproduction begins in the nuclei of host cells. Tissues infected and pathologically are also very similar to the nuclear polyhedrosis viruses. There are three major genetic types of granulosis viruses. Type 1 granulosis viruses described from the cabbage loop Trichoplacea knee only infects the midgut cells and subsequently the fat body cells because it does not infect the tracheal matrix or epidermis. Larvae may live longer than NPV infected insects. Type 2 GVs, granulosis viruses, first isolated from the codling moth, Sidia pulmonella, parallels NPV infections. Sidia pulmonella granulosis viruses. Sidia pulmonella granulosis viruses is a baculovirus granulovirus. It has a double-stranded DNA genome and 1,23,500 base pairs in length with 143 open reading frames. The virus forms small bodies called granules containing a single virion. GPGV is a virus of invertebrates, specifically Sidia pulmonella, the codling moth. CPGV is highly pathogenic. It is known as fast granulosis viruses, that is, one that will kill its host in the same instar as infection. Thus, it is frequently used as biological pesticide. Coming to the third one, type 3 known only from the western grape leaf skeletonizer called as Hericinia brillians, which infects only the midgut tissues. Advantages of microbial insecticides. The organisms used in the microbial insecticides are essentially non-toxic, non-pathogenic to wildlife, humans and other organisms, not closely related to the target pests. The safety offered by microbial insecticides is their greatest strength. The toxic action of microbial insecticides is often specific to a single group or species of insects, and this specificity means that most microbial insecticides do not directly affect beneficial insects, including predators or parasites of insects in treated areas. If necessary, most microbial insecticides can be used in conjunction with the synthetic chemical insecticides because in most cases the microbial product is not deactivated or damaged by residues of conventional insecticides because the residues present no hazards to humans or other animals. Microbial insecticides can be applied even when a crop almost ready for harvest. Disadvantages of microbial insecticides, because a single microbial insecticide is toxic to only a specific species or group of insects. Each application may control only a portion of the pests present in the field or garden or lawn. If other types of pests are present in the treated area, they will survive and may continue to cause damage. Host desiccation, that is the drying out, or exposure to ultraviolet radiation reduces the effectiveness of several types of microbial insecticides. Special formulations and storage procedures are necessary for some microbial pesticides. Apart from all these things, researchers are going on to develop the effective integrated pest management or microbial pesticides for the increased production of the pathogens which causes detrimental to the agriculture crop plants. So the research is open and the area is wide open, and the researchers are still going on to produce the effective biopesticides to control various types of diseases. And the war is continuously going on between the pesticide and the pest, okay, so that we can save the agriculture cropland. And if you save the agriculture cropland, definitely the human being will have the healthy life. So with this I conclude, and I sincerely thank the organizer for the given opportunity, and hope this talk would have given very good information on the biopesticides against various disease causing insects and pests and microbes. So I thank everyone for your patient listening. Thank you.